good morning to all. So now we are going to see about AIDS. AIDS means an acquired immunodeficiency syndrome. So what is AIDS? It's a disease that is caused by a HIV virus that causes AIDS. Disease limits the body's ability to fight the function, that is infection caused by another virus system. A person with AIDS has a very weak immune system. Um, there is no cure for this disease because AIDS is a killer disease. So, what is the next? What is the history of AIDS? In 1959, the scientists isolate what is believed to be the earliest known case of AIDS. The discovery suggests that the multitude of global AIDS viruses all shared a common African ancestor within the past 40 to 50 years. In 1978, gay men in the US and Sweden and the heterosexuals in Tanzania and Haiti began showing signs of what will later be called as AIDS. In 1972, the term AIDS, acquired immunodeficiency syndrome, is used for the first time on July 27th. It is coined on July 27th only. The next is structure of a HIV virus. So, what is the structure? Structure of the human immunodeficiency virus belongs to this. It looks like a coronavirus itself. Like it have glycoproteins, lipid membranes, RNA, capsid, matrix, and reverse transcriptase. So these are the formations of this HIV virus. And what is the HIV transmission? How it transmits? Common fluids that are a means of transmission: blood, semen, vaginal secretions, breast milks. These are the forms. That is blood where it mixes with other ingredients, other things like breast milk. Vaginal secretion is a fluid that secretes from a hormone, a vaginal excreta, and the semen that is produced by a human body, and a blood that runs over our whole body. So these are the fluids that are means of transmission. So in the stage 1, there are 4 stages of HIV virus from stage 1 up to stage 2. What is stage 1 means? Primary. Short flu-like illness occurs 1 to 6 weeks after infection. No symptoms at all. Infected person can infect other people too. And stages, asymptomatic. Last for an average of 10 years, this stage is free from symptoms. There may be swollen glands. The level of HIV in the blood drops to very low levels. That is, if there is a HIV infection, the blood drops to a very low levels of hemoglobin. Okay. HIV antibodies are detectable in the blood only. Then how it transmits in stage 3, that is symptomatic. So the symptoms are very mild. The immune system deteriorates. Emergence of opportunistic infections and cancers are also occurred during this period. That is during this stage of symptomatic AIDS. The immune system will get affected severely. But the symptoms are very mild only. So what is HIV? The immune system, that is the stage 4. The total immune system is destroyed and collapsed. The illness become more and more severe, leading to an AIDS diagnosis. Okay, what is the HIV cycle? How it forms and what is the cycle of it? Let's see. So this is the HIV and the fusion of HIV with the host cell surface. So what is a host cell? The blood or other fluids that transmits HIV. Okay, there are also some cell membranes. When that cell membranes are carriers who transmit HIV virus to other parts of the body and get infected. So the person who are infected by HIV virus, then he have no cure. So fusion of HIV to the host cell surface. So is just injecting it. Then what's the next one? HIV RNA. So it changes the RNA symptoms of the human body to whom it gets affected. So reverse transcriptase, integrase and other viral proteins enter the host cell. Then what is the next one? Pre-integration complex. That means what? Already the human body have some RNA. So that RNA will mixed together, that will combine together and form another form of substance, another form of RNA. So here they are shown it clearly. See, viral RNA, reverse transcriptase. So the RNA itself totally changing its shape, its attribute, everything. And integrase. So integrase means what? It is changed into another viral DNA. So new viral RNA will be formed. Okay, so the RNA will not listen to human's brain. So new viral RNA is used as genomic DNA and to make viral proteins. So it will make some viral proteins which will become, then it will become into a HIV virus. Okay, new viral RNA and proteins move to the cell surface and a new immature HIV forms. So here the immature HIV forms, and the next phase is virus is released. Viral protease cleaves new proteins to a mature and continuous virus. So this is known as a HIV virion, a mature virion. Understand? So what is the next one? Importance of early testing and diagnosis allows for early treatment to maintain and stabilize the immune system response. Decrease risk of HIV transmission from mother to newborn baby. So, allows for risk reduction education to reduce or eliminate high risk behavior. What they are saying is early treatment is used and stabilized to make the immune system more responsible. So, to decrease the risk of HIV transmission, the mother should be very careful while giving a birth to a newborn baby. Allows for risk reduction, education to reduce or eliminate high risk behavior, etc. So, what is HIV testing? Requires a blood or oral fluid sample. The test requires to detect the body's antibody response for HIV infection. The test does not detect any HIV virus. So the first main test to do to detect the infection of AIDS is ELISA. That is ELISA or EIA. That is reactive. If it is reactive, repeat EIA or ELISA. When it is again reactive, IFA test should be conducted. That is reactive. Then the HIV is positive. Then HIV will be positive. Okay. So thank you for listening to this. Let's see in the next video.